Okay, so in week 6, one of the tests that you'll do this day in lab that you'll actually see the results from is a test called the coagulase test. So this is not a test where you inoculate a medium and then wait for the bacteria to grow. This is a test that uses bacteria that have already grown. So the coagulase test is a test that's used to assay for a specific enzyme, which is an important virulence factor in staphylococci. As a matter of fact, those of you who go on in the biomedical field will perhaps never hear excuse me, staphylococci referred to very frequently as Staph aureus, Staph epidermidis, Staph saprophyticus, as we use here in the laboratory. You'll most often, in the medical context, hear them referred to as either coagulase positive or coagulase negative staphylococci, or sadly, methicillin resistant staphylococci. But that's another story. So what you're going to do today is to assay for this important virulence factor. So this test is done using rabbit plasma. So I've reconstituted a lot, and the TAs have reconstituted even more of this rabbit plasma. So the difference between plasma and serum is an important distinction. When blood is drawn by venipuncture, if that blood is drawn into an anticoagulant, such as sodium citrate or more likely heparin, the blood doesn't coagulate. It stays in the fluid phase. So at that point, you can separate the cellular component from the fluid component by centrifugation. The fluid component from that uncoagulated blood is referred to as plasma. That is, in fact, the fluid phase of all of our blood. If, however, blood is drawn with the idea of getting serum, which is frequently used for antibody tests, blood can be drawn without the use of an anticoagulant, in which case the blood forms a clot within the vessel that the blood is collected in. Then what can happen is that the clot, the cellular clot, can be centrifuged out, can be removed by centrifugation, and the fluid phase remaining then is referred to as serum. The difference between plasma and serum is that plasma maintains within its fluid all of the proteins necessary for clotting, fibrinogen, all the members of the clotting cascade. In serum, however, those proteins have been depleted. They've been used up in the clotting process. So it's important that we always use plasma in coagulase, because what we're actually assaying for is the ability of the bacterium to form a clot. So virulent staphylococci possess on their surface, and to some degree a secreted enzyme called coagulase, which is capable of triggering the clotting cascade in much the same way that we trigger the clotting cascade. This ends up being a virulence factor for staphylococci because it can help staph to hide from our immune system by quite literally cloaking themselves in fibrinogen and other members of our clotting cascade, so it makes them more difficult to recognize, perhaps more difficult to phagocytose and to kill. So coagulase is a very important virulence factor that staphylococci have. And so we're going to assay for the ability of 3 different species of staphylococci that we use here in the laboratory to produce the enzyme coagulase. So what you're going to do is, for each of the 3 for each of the 3 species of staphylococci, you're going to assay using coagulase. So each of you will take 1/2 of a milliliter or 500 microliters using a pipetteman like this. I suspect that many of you have already used a pipetteman like this. If you haven't, let myself or one of the TAs know, and we'll show you how to use these things. There's a second stop to a pipetteman like this, so it's important that you know how to use it and how it's properly set. This is currently set for 500 microliters or 1/2 milliliter. I'm going to draw out 1/2 of a milliliter of this rabbit plasma and put the rubber cap back in. And I'm going to put this into, and again, sterility is not important here. We're not going to culture anything. We're just looking for the presence or the absence of a clot. So I've added that 1/2 ml of rabbit plasma to that tube. And then I'm going to eject that pipette tip and set this pipette aside because I don't need it any longer. So what we'll do next is to use a sterile loop. So I have the flame going. I think it's outside of the picture. I'm going to sterilize this loop, and I'm only going to do one species right now to save on resources, and this is just for illustration. But I'm going to take one of the staphylococcal species. So you'll be provided with 3 different species, and I'm just going to take a pretty good slug of the bacteria just to pull through that. So you can see a pretty good chunk of the Staphylococcus on there. And now I'm going to go into this rabbit plasma, and I'm just going to work that Staphylococcus pellet until it is largely in solution in the serum. You can hopefully see a little bit of turbidity in the serum as this is going. I'm going to sterilize this loop before I set it down, because remember that we're dealing with a human pathogen here, and hopefully you'll identify which one of these species is most likely to be virulent based on the presence of coagulase. So I'm just going to gently mix that a little bit. And you can see a little bit of more turbidity to that plasma now that the staphylococci are mixed in there. Then it's always also very important that you label which one is which, because you'll be doing 3 different species. Then what you're going to do is, once all 3 of them are done, you're going to put them into a 37-degree water bath that is set up on the bench in the laboratory, and check them after an hour. If you still do not have one of the organisms testing positive, that is giving a clot, by 1 hour, just leave them in the water bath, and myself and the TAs will take them out later and put them in the refrigerator, and you can see the results when you come back for the next lab. But this is a good exercise to do first in the laboratory this week, because this test can take an hour to an hour and a half to occur. So if you do this one first, you have the best chance of seeing the results before you leave for the day. So that's the coagulase test. The next part of the video is going to be talking about tests that we did last week and some more inoculations for this week. So this is the end of this section of the video, but I'm going to knit this together with a video that I already did yesterday covering the rest of the tests. So yeah, obviously neither microbiology nor filmmaking are my forte. So bear with me, and hopefully by the end of the semester I'll get this down. But yep, I'll see you in just a few seconds on the other video. Good morning. So for the lab pre-video for week 6, first we want to deal with the tests that you inoculated for week 5. So we'll start first with the nitrate test, because this is the more complicated of the tests that you're going to do. Actually, it's the only test that has any level of complexity. So what you're going to do, you already inoculated a media called nitrate broth, which is very rich in potassium nitrate. The idea being that if, as bacteria grew in there and utilized the oxygen, if they were capable of switching their respiratory chain to utilize nitrate in anaerobic respiration, they would reduce that to nitrite. So in essence, what we're going to do is assay for nitrite ions. So the 2 chemicals that you're going to add here, appropriately enough named nitrate A and nitrate B, are a combination of sulfonilic acid and dimethylnaphthylamine. These reagents will give a bright red azo dye if nitrite ions are present. So this test is highly specific for nitrite ions. So the first thing that we're going to do is we're going to add 4 drops of nitrate reagent A. 1, 2, 3, 4. I'll go 5 just for good measure. That's the sulfonilic acid. I'm going to mix that a little bit, and then I'm going to add nitrate B, which is the dimethylnaphthylamine. I'm going to add about the same volume of that. 5 drops. 1, 2, 3, 4, 5. And I'm going to mix that a little bit and start and set it aside. In reality, what you can already see is a red color beginning to develop. So what that red color is indicating is the presence of nitrite ions. The media, as it began, was very rich in nitrates but very poor in nitrites. So this is uninoculated media. So we're going to use this as a control, just so you know that this red color doesn't just develop as a, just automatically in this media. So again, to this uninoculated control, I'm going to add 1, 2, 3, 4, 5 drops of nitrate A. Mix it slightly, and now 5 drops of nitrate B reagent. 1, 2, 3, 4, 5. Recap that and mix it up. So you can already see, even though it's not quite a fair comparison because I added the reagents earlier to this bacterium than to this uninoculated, but you saw how rapidly the red color developed. So this test for this particular organism on your left would be nitrate reductase positive. This is an organism that can anaerobically respire using nitrate as its terminal electron acceptor. So it reduced that nitrate to nitrite, but it took that nitrite no further. It could do nothing with that nitrite. So once this bacterium, this happens to be, well, I won't tell you. I'll let you be surprised by your bacteria. Once this bacteria utilized up oxygen in the environment, in its environment in this tube, it was then forced to switch to utilize nitrate, reducing that to nitrite in order to maintain respiration. Once nitrate was utilized in its entirety, this organism would likely have had to rely on fermentative metabolism. So doing fermentation to reduce, or excuse me, to reoxidize its electron carriers, NADH. But in any case, this is a definitive positive test for nitrate reductase. And you can see that in the nitrate broth, there was initially no nitrites. So we can set this aside. So I have inoculated other nitrate broth cultures with a consortium of soil bacterium, which is kind of a fancy pants way of saying I put some soil in here and let it grow. The idea being that soil microbes, remember we talked in lecture about the bacterium Paracoccus denitrificans. That wonderful little soil bacterium is a facultative anaerobe. It has the ability with its electron transport chain to reduce oxygen at the end of its electron transport chain when oxygen is available. But when oxygen is gone, it can switch over and reduce nitrate to nitrite. But it's not done there. When nitrate is utilized in its entirety, Paracoccus denitrificans can then reduce nitrite to nitric oxide, and then reduce nitric oxide to something like nitrous oxide, and even to to atmospheric dinitrogen. So there are other reduction processes that can occur in bacteria, and you could get a false negative. So that's what I'm hoping is going to occur with the soil bacterium. So this year, the TAs convinced me to see if we could capture any gas being produced. So they put in Durham fermentation vials in here. And unfortunately, I'm not seeing any production of gas. Maybe you will in yours. But let's do the nitrate test on this collection of soil bacteria. 1, 2, 3, 4, 5. Let's go 6. I unfortunately went to that school that said if a little is good, a lot is better. Doesn't really work. And then nitrate reagent B. 1, 2, 3, 4, 5, 6. So we are beginning to see some pink color here. So this is not not the outcome that I had hoped for. This suggests that what's occurred as these soil bacteria have grown in here is that they have reduced nitrate to nitrite, and they haven't yet, if they're ever able to, they haven't reduced nitrite to another form, such as ammonia, such as nitric oxide, such as atmospheric dinitrogen. So there has been nitrate reduction in here, as evidenced by this red color. Okay, let's imagine that this result had come out as I expected it to, in which there was no red color. The absence of red color could be due to two, so this is why the nitrate test is a little bit confusing. A negative result like this, after adding nitrate A and B, as I did in this uninoculated culture, could indicate that the bacterium was simply incapable of reducing nitrate to nitrite. That's one possibility. There's another possibility to your interpretation of a negative nitrate reduction test, is that the bacteria may have reduced nitrate to nitrite and then further reduced nitrite to some other form, possibly a gaseous form. But remember that these two reagents, nitrates A and B reagents, are going to create that azo dye only in the presence of nitrite. They're exquisitely sensitive for nitrite. So if the bacteria have already used up the nitrates and the nitrites, you're going to get a false negative. So the way that you determine whether the false negative is that you will now take a toothpick. This is very high tech. And some zinc dust. You're going to take the toothpick, put that into the zinc dust. You don't need a lot. You just want to make the end of that toothpick kind of dirty with zinc dust. Zinc is a very strong reducing agent. So when we add this toothpick to that mixture, if there are any nitrate molecules left in there, the zinc will reduce them from nitrate to nitrite. And since we've already added the reagents, nitrate A and nitrate B, the reduction by zinc of nitrate to nitrite should give us a red color. This is obviously not my morning. Well, it's slowly coming up. So you'll let this sit for about 15 or 20 minutes, but you can already kind of see a red color developing in here. So what's happening, what this means is that there was still nitrate present in that media that's now being reduced to nitrites by the presence of the zinc ions. That red color now indicates that this bacterium, I know there's no bacterium in here, this is my uninoculated control. Let's pretend there were bacteria growing in here. What this red color means after the addition of zinc means that this was a true negative. This, if you did not get a color here with the zinc, that would mean that the bacteria in that culture had reduced nitrate beyond nitrite to something such as ammonia or nitrogen gas, something that this test will not determine. So hopefully what you'll find as you do your nitrate tests is a collection of bacteria that are truly nitrate positive, nitrate reductase positive, some that are nitrate negative, and some that have reduced nitrate to some form beyond nitrite. So please read the PDF posted on the Blackboard site about the nitrate test. That will make it, hopefully it will make it less confusing than I feel like I just made it. Okay. So the other tests that you did in week 5 while you were in the laboratory, how do we interpret them? None of the other tests that you did require the addition of any reagents. These are simple observational tests. So you inoculated several bacteria with media like this lactose tube broth. So this is an uninoculated lactose tube. It's at a neutral pH, so the pH indicator is right around kind of a reddish color, not bright red. And its derm fermentation vial is filled with fluid and there are no gas bubbles. A positive reaction when you've inoculated a broth for carbohydrate fermentation, so in this case lactose, this organism on the left would be a positive reaction for an organism that's capable of fermenting lactose, and it's also capable of fermenting lactose with the production of gas. A portion of the gas produced by this bacterium during fermentation of lactose was captured here, probably a mixture of hydrogen gas and CO2, almost certainly. So that would be a positive reaction. A negative reaction, in a negative reaction, you can tell that the bacteria actually grew because of the turbidity in this tube on the left. Remember, the tube on the right is uninoculated. This, because it hasn't changed the pH to an acid condition, means that the bacteria did not create acidic fermentation products. So this bacterium would be negative for the ability to ferment lactose. There is a little bit of a pH change in the media inside the derm fermentation vial. If you see something like that, but the rest of the media remains neutral or alkaline, that's a negative. You consider that a negative reaction. And again, this particular bacterium utilized this other carbohydrate, sorbitol. It fermented that sorbitol very well and produced gas in there. And so sorbitol is an important diagnostic test used in microbiology. So the interpretations of your carbohydrate fermentation tests are very simple and very straightforward. The same with the other test that you inoculated. This was the urease broth. So you inoculated 3 different urease broths, and this is what they look like uninoculated. I didn't pull a truly uninoculated one from the refrigerator, and I should have. But what you see here is 2 negatives on the outside flanking a positive urease organism in the middle. This media has a very rich source of urea within it, that 1 carbon molecule with 2 amine groups associated with it. The media looks like this to begin with. The pH is around 6.8, and the pH indicator is this color. If a bacterium possesses the enzyme urease, it will break down urea as a nitrogen source, and it releases both carbon dioxide and ammonia. And especially the ammonia leads to an alkaline reaction. So what you're seeing is that this particular pH indicator, phenol red, the same pH indicator that turns yellow at acid conditions, turns a brighter red, a fuchsia color, under an alkaline reaction. So when you see this reaction, this color, this means that this bacterium has broken down urea and released carbon dioxide and ammonia. If you want to know the actual reaction, you chemistry geeks out there, urea can be broken down by urease into carbon dioxide and ammonia. And again, please read the PDF from the American Society for Microbiology that I posted on the Blackboard site. So that's all you have to do in terms of interpreting your tests from last week. This week in the laboratory, you're actually going to just be inoculating a lot of different media. We're getting close to doing the unknowns, so this week is a big push in terms of inoculating different media. And I'll explain more about the basis of these media next week when we actually have the results for them. And for the most part, you're going to be inoculating plates, streaking for isolated colonies, so it'll be good practice for your streaking for isolated colony techniques. The only media that you need to inoculate that's not in a plate form is at least one medium that you've already inoculated during the first week, just as a demonstration of slant media. But this week, you're going to deal with triple sugar iron agar again, these slants. This particular slant, it's important that you inoculate it properly, because you want to have bacteria growing not only on the surface of the agar slant, but down in the deep portion or the butt portion of this slant. So what you're going to do is, with a sterile needle, you're going to take the prescribed bacteria. This is Salmonella enterica. So, and this points out the fact that you are going to be dealing with human pathogens at this point. Pretty frank, pretty severe human pathogens. I'm going to take my sterile loop and I'm just going to touch one colony, close that. Remember, the good technique is I never set that cap down. I keep it in the pinky of my finger. Pass that through the flame. And now what I'm going to do with shaky hands is to rub that back and forth across the entire surface of that slant a couple of times. And then I'm going to stab down all the way to the bottom of the tube, at least once. Sometimes you may want to do it a second time. Pass that through the flame again, recap the tube, sterilize your loop, your needle. Label this culture, and it goes into the incubator. What you're doing is setting up a biphasic type of culture where the bacteria, in this case Salmonella, are going to grow aerobically on the surface. They're going to be in contact with oxygen at all times. The Salmonella that you have inoculated down into the agar deep portion will utilize the oxygen in that environment quite readily, but oxygen doesn't diffuse very well in fluids, and even worse perhaps in agar. So an anaerobic condition is established in the base here, and so bacteria will alter their biochemistry, alter their metabolism under anaerobic conditions, so we can actually assay for multiple biochemical capabilities of bacteria, both aerobically and anaerobically. And we'll talk more about the specifics of this media. I'm going to post under week 6's folder, along with this video, some PDFs on one of which will deal with the triple sugar iron agar. This is a complex media. There's a lot of information that can be gained from this, so it's going to be very important for you in terms of your unknowns and in terms of the lab practical that you understand this media very, very well. The only other media that, other than plate media, where you're streaking for isolated colonies in week 6, is inoculation of another slant media for another amino acid test for phenylalanine deaminase. And again, you're going to use your best sterile technique to pick a colony. I'm going to actually go back into that very same colony of Salmonella enterica. Take the cap to stab that all the way down through the butt, through the slant portion. You're welcome to. I don't know if that's helpful, because we're going to assay after fall break for the ability of Salmonella enterica and some other bacteria to deaminate the amino acid phenylalanine. And again, I'm putting up a PDF to explain this test in more detail and in clearer terms than I am. That's going to be found very shortly in the Blackboard site. So I think that's really all for this week. And yeah, I'll see you in the lab.